very brief because Anuj is one of us. So it's a pleasure to introduce Anuj Mubayi. He has been working with us on and off, but he was at ASU before for a new venture in his life. He'll tell us a little bit more about it. But he's been very actively working with us, indirectly at least through folks and his own work. And he's done some remarkably interesting work on SARS-CoV-2. His background is in applied mathematics. And I must say, Anuj has got an excellent record in terms of mentoring students. Many of his students have gone on to find very good positions in other places. If I remember right, one of the postdocs in Allison's group is also your student, Anuj, is it correct?
Yeah.
So Abhishek is so we have connections here and in Allison's group. So thank you very much, Anuj. Looking forward to your talk.
Thank you, Madhav, very much. First of all, inviting me for this presentation so that I could share with you all my background, my experiences, but also making me part of this big community where I'm learning every time I was part of the seminar or some discussion or some papers that comes out of the group. So thank you very much. It's very kind of you all to invite me for this presentation. Well, I'm currently at PRECISIONheor, which is a health economics company. I do many of those things that I used to do, and they give me a kind of a free hand in many of these things. In fact, we are working on COVID, but because of these working with the clients, it is difficult to present those things until we get clearance on those projects. So I thought of what to present today. So I'm presenting today is actually one of my old and very dearest research topics on neglected vector-borne diseases. And I'm, as Madhav was mentioning, I'm an applied mathematician by training. So much of the work is on how you study the complexity which are inherent in this type of disease system. I'm also currently part of Illinois State University, and I'm teaching a biomathematics course there, as well as College of Health Solution at Arizona State University. So briefly, neglected tropical disease, what are those ones? What is critical aspects that we care from a modeling and from the practical ground level perspectives? What are scales in it? What scales we talk about? And then what do I mean by saying ecological complexity when we try to capture the risk associated with this neglected vector-borne diseases? For example, there are multiple host species, there are vector preferences, vectors are like mosquitoes and malaria, for example, and spatial heterogeneities, the people from one location versus people in other location. And then I will talk about two examples from my research. It's on leishmaniasis, visceral leishmaniasis in particularly, and Trypanosoma cruzi, which is a pathogen for Chagas disease. Why we talk about NTDs? Well, with COVID has changed quite drastically. Before COVID, there was already quite differences in the way the people were having the resources for commonly directly transmitted disease versus neglected tropical diseases. But after COVID, this gap has increased drastically. There was already a huge gap before COVID, but during COVID, this gap of availability of the resources and practical, whether it is a treatment, whether it is an intervention of any sort, whether it is resources to control it, has become down drastically. And this is a major concern in near future. If we will not focus on it, there will be a huge challenges to the society as a whole. So when I talk about neglected tropical disease, vector-borne diseases are kind of a core of these diseases. For example, malaria we all heard, but some of the unknown diseases are leishmaniasis, Chagas disease, and these are, or lymphatic filariasis. These are caused by certain pathogen, but they are not directly transmitted. They are transmitted by some vector species. For example, leishmaniasis is spread by sandflies. And there are different hosts which are involved in it. And they could be anthropomorphic or zoonotic. Anthropomorphic means humans are the only carrier, whereas zoonotic means that animals could be, or mammals could be part of the cycle of the transmission. It disproportionately infect many of the poor countries and tropical regions. As you can see in this map, shows very clearly what regions are affected. So in 2012, WHO tried to focus on neglected tropical disease with the help from Gate Foundations and 10 big pharmaceutical companies, which were ready to put some resources to control of many of these diseases. So basically, neglected word means the resources are very limited. There's not much efforts by pharmaceutical companies. These are hundreds of years old disease. So at least 100 years when we say we recognize it as a disease 100 years ago, but it could be more older than that. And it's affecting billions of people in the world. Primarily the poor and developing countries are affected by them. So they are divided into somewhat micro and macro. There are 21 listed by WHO as a neglected tropical disease. Of course, disease as a whole, we talk about pathogens and viruses and protozoa bacteria, but then also in the same list, there is snake bite, also part of this neglected tropical disease. People die from snake bites in some of the developing countries. But my focus in my work has been on protozoa diseases, Chagas and leishmaniasis. I've worked somewhat on lymphatic filariasis as well, which is helminths, worm diseases. And they are modeled in a very different way, microparasite and macroparasites. Microparasites are smaller and macros are worm diseases, which are bigger in size, roughly. And there are other characteristics which makes it different between. What I do in my research is complexity involved in it. How do we understand that what aspects of the systems is driving the prevalence of the disease or dynamics of the disease in general? The disease system has incredible diversity, with whether we are talking about different types of pathogen species, host, vectors species. So these all contribute along with the environment around it, causes different types of transmission cycle to exist and the diversity in the disease system. In order to make some interesting aspects on my research, I want to first give a few examples, which are kind of a counterintuitive example to control NTD. During NTD control, NTD stands for neglected tropical disease. There has some things that people saw which was unknown earlier. So for example, visceral leishmaniasis in Brazil is spread among humans, of course, and it's a major public health concern in Brazil. But domestic dogs are primary reservoirs. So much of the effort the Brazil has focused on is to control domestic dogs in the community. And one thing what they do as a control is to mass eliminate seropositive dogs. And they think that by doing something like that, they would be able to decrease the prevalence of zoonotic visceral leishmaniasis. However, from 1990 to 1997, around 176,000 seropositive dogs were killed, but the disease didn't reduce. And that was the challenge in 1990s, why the prevalence didn't decrease in spite of so many killings of these dogs, which were the primary reservoir host for it. Well, in fact, what they found is that the number of human cases increases because sandflies were now biting humans because they were not having those domestic dogs as a reservoir host, those that were killed during the mass elimination. So this is a very good example which shows that a host diversity, how it changes the infection prevalence in a not a simple way. They killed many dogs because of this, but it was a counterintuitive examples, and the disease prevalence in fact increased at the end of the 90s, in 1990s. Second example, this one is a study from India, which is control of malaria in India. And there's a study in 2016, which talks about that all the efforts by Indian governments for Ministry of Health were focusing on human indoor residual spraying for malaria control. But it seems like many of the villages in India, especially the study talks about in Orissa is a state in India. They have these cattle shed right next to their houses. And many of these species of mosquitoes are zoophilic. That means they prefer feeding on kettles. And they rest in the cattle sheds and they feed on both cows and humans. And if you are focusing only on human dwellings, that controls is less effective, and you have to even consider kettle sheds. But you have to understand this study in particularly found that the mosquitoes are zoophilic behavior. That means they prefer cows as well, or kettles. So in fact, what they did was they did an experiment, and they imitated a temporary kettle sheds around the villages. And once in the evening, they take these kettles around the villages to put it in those kettle sheds. And they found that the disease incidence drastically dropped in those villages. And so just shifting the kettle sheds from next to their houses to the periphery of the village resulted in drastic decrease in the incidence of the malaria in those villages. So what this is showing is the changes in the dynamics of the density of the host species in the environment can drastically impact the prevalence or incidence of the infection in the community. So beautiful example, this one. And now they are focusing on kettle shed, in fact, for indoor residual spraying as well. This third example actually is of a disease, visceral leishmaniasis, which is also known as Kala-Azar in India. It's a life-threatening disease. People die from it, and if they don't go for treatment, even with the treatment, there's quite a significant mortality. It is spread by the sandflies, insect sandflies. This visceral leishmaniasis is the second largest parasite killer in the world, responsible for 30,000 deaths worldwide. So, of course, this is the disease well known, 100 years old disease. But then the disease post-Kala-Azar dermal leishmaniasis is a sequel of disease, same parasite, same way it is transmitted, but it's only 1 to 5% of successfully leishmaniasis treated patients develop this thing. So why it happens, it is not very clear, but it was from a clinical perspective was described in 1922. But it's a sequel of the disease, and we are talking about humans. Some humans are showing only visceral leishmaniasis treated successfully, but there are some fraction who are treated for visceral leishmaniasis get this PKDL disease. Now, in 1980s, there was a huge DDT spray program for malaria killing that brought down even the sandflies drastically. And that resulted in literally not having visceral leishmaniasis in the community. However, in 1991, this leishmaniasis came back. If you see in this picture, in 1991, it came back in spite of drastically reducing sandfly population by DDT spraying. DDT is insecticide that was extensively sprayed for malaria control in 1980s. So why did it came back? Later on, it was found came back because they were not considering these cases of PKDL in their surveillance system. And although there were few of cases, but they were drastic in initiating the outbreak of the disease. So what it shows is the host species. In the previous example, it was the kettles were the host. But in this example, it's a different stage of the disease. Those type of people can also cause the infection. So the host could be same, but the infection stages in the same host species can initiate the outbreak if it is not properly considered in the disease system. So these three examples, one on dog as a primary reservoirs in Brazil for zoonotic visceral leishmaniasis, and kettles in the malaria in India, and PKDL cases, which are human cases, but of a different PKDL patient do not die, by the way, and visceral leishmaniasis die from the disease. So these different types of individuals, human individuals, can initiate an outbreak. So what I'm discussing here is something that has been recently recognized as that in order to study a disease system, especially of vector-borne diseases, we have to consider something called as integrated human and animal health approach, which also takes into account socio-ecological system, and it is referred as in the literature as One Health approach. And malaria is a very good examples. The malaria that I was showing just now, the livestock can be used as an interventions. for control of malaria because you can divert the bites from the human to kettles. However, there is a problem with this approach also, is that the livestock, if you put a lot of livestock and the vector is start to bite more livestock, then you are counterintuitively, you are also increasing the density of the vectors in the community, and therefore you may increase the infection incidence in the humans. So there has to be a very light balance of how much livestock you use as an interventions and what is the preference for the vector towards this livestock. So there has to be a balance we have to do. Otherwise, we may think that we are doing good, but overall there might be a negative impact. So the factors that from a mathematical modeling that what we consider, basically what we do as an applied mathematics is we talk about threshold behavior in the system. When can things tip off? There's a beautiful book, The Tipping Point by Malcolm Gladwell, in which he talks about a real life scenario, like for examples, roids. Roids sometimes becomes a big, and some other times it is not really a big issue. For example, Arab Springs. Somehow Arab Spring spread. Some in Tunisia there was a local problem, but that becomes into a big Arab Springs, whereas many times the similar type of problem in different locations doesn't becomes like a big thing. So this Malcolm Gladwell in Tipping Point book is talking about real life examples without considering the mathematical models into it. But we do a similar thing in applied mathematics. We talk about bifurcation analysis, and we rigorously study the components of the models and how that goes into when the system tips to a different state. It is less of a prediction than more of a understanding of the mechanism. And it can be very easily understood from an epidemiological context. The vector-borne diseases that I'm discussing right now has, of course, host, vector, and pathogens that are coming together in a certain way so that the pathogen can spread in the environment, in a certain environment. But there are certain things that are very critical in it. For example, different type of host species. What is the strength of each host species? Competence. That means, are they able, some hosts are able transmit the infection good, where some are not. Social behavior of the host. Vectors are important because vector feeding preference, whether they feed on kettles, whether they feed on human more. So those things are very critical in the dynamics of this vector-borne diseases. And of course, the pathogens characteristics becomes very critical. One more thing which becomes very important in this is the transmission cycles. For example, how the transmission is occurring, whether it is occurring as a vertical transmission from mothers to babies, or oral transmission. That means by mistakes, animals are eating the insects, and insects which are infected with the pathogens are transmitting it to the host because they are by mistake, they are eating it. So oral consumption becomes very critical in some of the disease. As we all know, and I just give a brief outline to make my point more clearer, we all know how to model incidence rate, and many different assumptions goes into that. One thing which is there in these assumption in the directly transmitted disease is a contact rate, C, and then P is a probability of transmission given a contact, given a single contact. And so how do you model the contact rate? Well, one way to model is taking contact rate as constant. If you take it constant, you get something what we referred as a frequency dependent transmission rate or standard incidence. But if you take a contact rate as of linearly depending on population size, then we get something called as a mass action. So the contact rate is linearly increased in the mass actions with the population size. So these two aspects are more commonly used in epidemic models. But if you have an indirect transmission, that means through the vector, then things becomes a little complex and you need to model it in a more complex way. For example, if you are interested in infected host transmitting it to the vector, which is, let's say, mosquitoes, through the bite, then you have to know the maximum number of possible bites per day by a single vector. You have to know the susceptible vectors, how many of those susceptible vectors are there. And that product will give you a number of total bites per day by all susceptible vectors. And if you multiply by proportion of infected host, then this product, the first three terms product, will give you fraction of bites or the number of bites that are all the susceptible vectors that are on infected host. But not all bites are able to transfer the infection. Some bites will successfully transfer, some will not. So you need a probability of successfully transmitting the parasite given one bite. So this is the way you model it, and that gives you a total incidence or total number of new cases that are generated by single host in the vector population. But if you talk about from vector to human, this is little different, because now you want to know how many vectors are landing on a particular host. So in that case, again, you need a number of bytes, but then you also need number of vectors landing on a one host. And that would give you the number of received by one host from all the vectors landing on the same host. And then you need a total number of susceptible hosts, of course, and the same way, number of bytes per day received by all susceptible hosts, proportion of those bytes that are on the infected host, and probability of successful transmission. So this is the very critical in order to understand how we model the biting or the transfer of infection, which is not same as modeling it in the directly transmitted disease. That's the important aspect. And if you simplify this thing in new infection in host by infected vector, then you get what you are seeing it in the right time. One thing to note is the denominator in both the terms is host, is not vector here. In directly transmitted disease, it's the same population that you are talking about. You are choosing infected person from the same population here. But in the vector-borne diseases, it's vector divided by the host here. This is a very critical aspect of it. So I published a book chapter a few years ago, and there I talked about what assumptions that goes in modeling these vector-borne diseases, and that could lead to a contradictory results. And basically, your assumptions could be whether abundance of host is there for vector feeding, or very few hosts are there, or like there is lots of vectors are there. So it depends on what assumptions that you are taking. And the incidence term is changed based on how do we assume that the vector is abundance or a host is abundance. And when you change these incidence term based on the assumption in the model, the reproduction number also changes, and therefore the estimates of the reproduction numbers could be very varied. So sometimes the studies talked about different reproduction numbers of the same disease, of the same region, but they have a different estimate just because their assumptions probably are different in those regions. Okay, so let's see how we model the host and vectors. There's some characteristics which are very critical is vector preference. There are different types of host species. You could have cattle, you could have human host, but then there could be that the vector could choose infected over susceptible due to some evolutionary aspects that vector could change their behaviors. And how do you model that is by this, there is a preference here in the denominator rather than a total population size of the host. We are taking some preference for the infected compartment. Alpha V is a preference, and for susceptible, there is no preference, basically. So denominators changes from the mixing population changes because now vector are preferring more the infected place. Host competence is also very critical. Host competence means ability for a host to transfer the infection successfully, can sustain it, can receive it. All these things makes it a competent host. Now, before we talk about competent hosts, there are many hosts, many different type of hosts that we talk about. Some are accidental hosts. They get the infection, but in general, we all have many viruses, pathogens in our body, but not affecting from the clinical perspective. Those are called accidental host, a host that gets an infection without impacting the host itself. Incidental host is the host can get infected, but cannot able to transfer back. If there is a second bite on an infected host, that cannot. And primary host is the host can get infected, and in the host, the pathogen develops over certain stages and become more stronger in transferring. Whereas reservoir host is they harbor the pathogens for long period of time without impacting the host. The pathogen doesn't cause any clinical symptoms and can have pathogens for long period of time and can transfer also very easily. So those are reservoirs. So there is a minor subtle difference between all these terminology that we talk about host. So what typically happens is in these complex models of vector-borne diseases, level of infection in the prevalence depends on right balance of vector preference, host abundance, and host competence. So all these balance has to be in a right way to know the level of infection in the community. So first study that I will talk about is on visceral leishmaniasis. This recent study was published in Royal Society Open Science. Here, what we are doing is to investigate system complexity, how do we model it, and how do we study system complexity in the visceral leishmaniasis on the threshold behavior, threshold for elimination, threshold for control of the disease, and also the prevalence of the infection. So the type of heterogeneity that we are talking about, we have host and vector. We have biting heterogeneity because some host vector bite, even in the same population, vector bite in a very different ways. Some hosts get more wide, maybe because of ecological aspects, because of vicinity with the habitat conditions. So we have incorporated biting heterogeneity into it. We have also have a reservoirs in this models we are talking about. Reservoir could be kettles or could be livestock in general. Reservoir heterogeneity, we have talked about reservoirs who are a good transmitter of infection and reservoir which are bad transmitter of infection, and a biting preference, how they bite. And also we have incorporated spatial heterogeneity, movement of people between the space. So all these factors makes this system very complex to understand it. And we have tried to attempt it to describe it very rigorously using mathematical tools or bifurcation analysis. The life cycle of leishmania has many reservoirs. It is 100-year-old disease, and we have not able to control it yet. Why is it the factors? And we only focus many times on humans, but there has been a recent studies in actually 2013 study, which takes the blood meals from these different hosts like cattle, goat, buffalo, rats, chicken, and they have found the incidence in many of these host species, different host species. So it seems like that vector is biting many different hosts, and there is a potential for infection spread through that. So we constructed this minimal model. Again, the goal is to investigate the threshold behavior in the presence of multiple complexity associated with it. So we talked about minimal models that we can understand, comprehend it. We don't want to exhaustive model, but we want a minimal model to understand it. So we took multiple hosts, 2 reservoirs, 1 humans, and 1 sandfly populations. And we talked about multiple patches. So patches are connected through the movement of the people. Then in each patch, we defined these interactions of these populations. For example, there's a human population, there is a sandfly population, there's a reservoir A, and there's a reservoir B. Reservoir A is animal reservoir, and animal maybe cows and goats, for examples. And also important is the term which is post-Kala-Dermal leishmania, which is sequel of the disease K. Here is the sequel of the disease, which is post-Kala. So it has many complexity, and we wanted to address what conditions on the model parameters which have biological meanings that the disease prevalence or the threshold behavior would change. One other things which we have incorporated also is a biting heterogeneity. So number of bites in mth individuals is Poisson distributed that we assumed in the models, and with a mean of certain thing where the mean itself is gamma distributed. So that is capturing the variation that has been observed in the field, and these distribution describe that heterogeneity quite well. If you do some calculations, and in the paper we did describe it in detail, the incidence is not a typical way of modeling it. The transmission term is not a typical way. It's actually a very complex way of modeling it. It has a logarithmic. This L stands for host species and V stands for vector. So in host species, the transmission terms is actually very complex, assuming all those heterogeneity that I talked about in the previous slide, as well as biting heterogeneity. And the vector heterogeneity is captured by this transmission term. On top of it, if I incorporate the movement or spatial aspects, human mobility, this transmission term becomes more complex, and the movement we are talking about is a short-term movement, not a long-term. So in short-term movement, you talk about resident time, that fraction of time of the daily time that people are spending in the different patches, for example. Then this transmission rate becomes more complex, but takes into account the movement of different host species. For example, a reservoir host is A and B here, and human hosts are, and you can incorporate by these two parameters that we are incorporating, delta ij and rho ij, which is capturing the movement between the patches. So if you have this transmission dynamic, the first question comes, there's so much of heterogeneity in this model. How do we study and how do we make sense out of the results? So we do a standard bifurcation in it. And first result, what we found is that this ROS is the complete reproduction number. Reproduction numbers, as we all know, describes the disease persistence or eliminations. Well, in this model, each patch's reproduction numbers are less than 1, but the overall reproduction numbers can be. So you can think about disease being controlled in each of the patches separately. But when we take the whole system where the movement of the people are also there, the disease will persist in those ones. Our reproduction number can be greater than 1. So we have seen the scenarios in our work. So one thing to here, what we have done, reproduction number is less interesting here in this vector-borne models than sandfly density threshold. So instead of reproduction number, we use a sandfly density threshold. Why it is important? Because sandfly density is controlled by insecticide spraying. And therefore, we want to know reproduction number is difficult to measure practically in the field in the vector-borne diseases. So sandfly density is what is used as a threshold quantity for understanding this disease. So one of the results suggests sandfly density threshold for VL elimination will be lower considering spatial structure. If you consider spatial structures, heterogeneity, then movement of the host and reservoirs, then the threshold levels are much lower. Threshold for VL control is much lower in the spatial model. So sometimes spatial structure is important, and if you do not consider, you would have a wrong threshold, basically, of VL elimination. So that could be the reason why visceral leishmaniasis may not be able to be controlled in India, for example, for so long. We also find that biting heterogeneity, if you increase the biting heterogeneity, it increases the threshold. So, okay, before I go into biting heterogeneity, one thing we have found in this is a backward bifurcation occurs. That means the disease if the backward bifurcation doesn't exist, then the disease has a clear-cut threshold phenomena at 1. R not less than 1, the disease can be controlled. R not greater than 1, not controlled. But if the backward bifurcation occurs, then there is much lower than 1 threshold conditions exist. And so backward bifurcation occur because of the two aspects in the models, disease-induced death rates and the movement rates. And that is a very interesting aspect that we have found in the model. Not only that, this threshold behavior increases in biting heterogeneity, the more variability in the biting characteristics on the host for the vectors, the threshold decreases over time. Threshold decreases as the biting heterogeneity increases. So you see the vertex of the parabola, this decreases, this vertex decreases with increase in biting heterogeneity. That is showing the threshold. So in order to control a disease, we need to make this density of the sandfly less than this quantity in order to decrease the disease, because then this is stable. But if it is density is bigger, then you could have a condition whether, of course, disease can be controlled, but if you are above this vertex, that this disease could be the endemic state is stable, therefore the disease will establish itself. So this vertex is very critical, and that describes the critical sandfly density rate to talk about. So the overall, the gist of the story is the biting heterogeneity, the variation, the larger variation in biting increases the sandfly threshold. So this sandfly threshold is increases. So this is increasing and lowers the infection. So this y-axis is a human prevalence, and it is lowering the rate of the human infection threshold. So very important aspects that we are learning from this model. Second thing, what we have found is if you have patches which are similar in characteristic, patches similar in means they have a same demographic and epidemiological characteristic as the other patches, then regardless of the connection between it or coupling or the movement rate in it, the infection prevalence or threshold behavior will be similar, as you are seeing in the left-hand side. Both the patches will have a same threshold behavior. But if you increase the dissimilarities, if the population characteristics will be different, quite different, then we see that the threshold behaviors are very different. So one of the way we are talking about dissimilarity is by taking a human birth rate higher in patch 1 than in patch 2. Okay, so in that case, what we are saying is sandfly density threshold for visceral leishmaniasis elimination will be lower if you incorporate spatial heterogeneity. So that's an interesting point also. The spatial heterogeneity decreases the sandfly density threshold. Now, another thing that we are interested in knowing is also integral stability or measure of resilience of the system. How resilient is our system? And basically, the overall idea is, the gist of it is, we are talking about basing of attractions in the backward bifurcation region. There are two equilibrium that are multi-stable system. This is a multi-stable system in the same parameter regimes. It depends on initial condition, which stable equilibrium the system will go into. But the basin of attraction, with respect to the parameters, if you perturb the parameters, how the basis of attraction squeezes or expand. Integral stability talks about that. And it basically gives you the resilience of the system, how resilient the system is to the perturbation of the parameters. So just to talk about the gist of the result is that as the reproduction number increases means you are perturbing the parameters, because reproduction number is a make of a lot of parameters. So as your reproduction number changes, there are so many moving parts in the system that it's very difficult to talk about changes in the perturbation. So reproduction number is the best way to change doing the perturbation. Integral stability changes for both the equilibriums, and it turns out in the multi-host model, the last bullet, disease-free equilibrium is more resilient when dead-end hosts are there. The reservoir hosts are the dead hosts than in their endemic equilibrium. So if there is a dead-end host in the system, the disease will most likely to die out. So this is about the threshold behavior. The second study here, we talk about scales here and how these scales. So very quickly, I will talk about this two-strain sylvatic transmission of Trypanosoma cruzi. Now here also, this is very complex pathogen transmission occurs by a vector species called as Triatoma. This is also called as kissing bug. The parasites is called as a Trypanosoma cruzi, which causes a Chagas disease in humans, by the way. it has multiple strains that are circulates in it. And there are many reservoirs of it, sylvatic reservoirs, such as raccoons, possum, wood rats, and so on and so forth. We are talking about in US as a wildlife disease. Why? Because those are the reservoir that harbor the infections, and the infection can spill over to human from time to time. So we need to understand what is happening in the sylvatic host. So humans are rare in US, but it happens, the infection that causes the Chagas disease. But the prevalence in the wild host species, sylvatic hosts, raccoons and possums, and wood rats are quite high, very high. So the characteristics of this Trypanosoma cruzi infection is multiple host, multiple vector species. Vector triatoma has different species that are circulating the infection. The transmission pathways are also very different. So just to summarize it, of course, to the vector, the infection is transmitted through the bites. To the host, the infection is transmitted in a multiple way, actually in three ways: through the biting, but also through the oral consumption. That means animals are eating the bugs by mistakes and getting infected. That's an oral consumption. And a vertical transmission, that means mothers to babies. So these are the ways that it can be transferred, three different ways. And in fact, there are two strains in US that causes this infection to spread: type 1 and type 2A strains. The interesting part is in Mexico, it is a very dominant strain. It causes Chagas disease. A lot of people die from the Chagas disease. But in US, it doesn't. So we model this Texas region, which is consists of three broadly speaking regions. This color coding is due to the cycle, distinct cycle in which specific type of host, specific type of vector species are present, and the strain. So for example, there is number 3, which is a US, primarily US, is the region where possum and raccoons are host species, and the species of the vectors are very particular. Whereas in Mexico, wood rats are there, and there is a different species. And in 2, which is a Texas region, these two cycles come together and interact. And the question that we were asking is the pathogenic strain in Mexico, which causes the Chagas disease, can take over and become common in US or not? What are the conditions under which these Mexican strain becomes common? So type 1, which is in Mexico, becomes or more dominant in the US, type 2. So we consider simple model: one host, one vector, two strain model, and three transmission pathways. Three transmission pathways because we have biting, oral consumption, and vertical transmission. And the system becomes a combination of host vector infection dynamics, but also predator prey types of dynamics. Why predator prey? Because oral consumption is one way of which the host is becoming infected. So predator prey and host vector dynamics comes together. And so what we found is the host transmission along with vertical. So let's talk about, so we found a bifurcation region in which one strain could be possible or both the strain could be possible. In fact, in this type of model, both strain is not possible in the same region. Only one strain can exist. So E1 stands for strain 1 existing, E2 stands for strain 2 existing, and E0 stands for none of the two strain exist, no infection exists. And there is a bifurcation on which we can actually plot a specific region on which these infections are persisting of each strain. So overall, the gist of the story was that adaptation to vertical transmission. So remember that type 1 is spread by only biting. Type 2 is spread by biting and vertical transmission, but oral transmission was somewhat not sure. So in order for a strain type 1 to take over the type 2, that means Mexican strain to become dominant in US, that it has to be adapted by over oral transmission. The host should be able to get infected in sufficient number by eating infected vectors. So this is a beautiful work that has been actually sequels of work has been published in many tropical disease journals also. So I want to thank at this point and stop here. I don't want to bore, but I think the whole story that I presented today was role of complexity that are inherent in this vector-borne diseases or neglected vector-borne diseases to study the threshold behavior. So thank you very much for having me.
Thank you very much, Anuj. Excellent talk. Very, very broad talk. I think it fits the interests of the expeditions project very nicely, and our institute's interest, in fact.
Well, one thing I want to add, Madhav, is also that I'm working on COVID aspects also, but I wanted to present this my own research focus, but I've been also working on airplane policies or boarding policies or deplaning policies, how that COVID has impacted that policy and what could be the right way to understand infection spread in the airplanes, for examples.
This is good, Anuj. I think we're all a little tired of COVID at this point of
I
time.
know that is why I present.
The COVID fatigue right now. So something different is absolutely welcome right now.
Anuj, can I just ask, have you published anything on that airplane work, or that's still stuff you're working on now?
Yeah, it is published actually. We have been doing it since 2015 when there was Ebola outbreak. So we have been publishing continuously. Recently, we actually talked about whether leaving the middle seat vacant makes any difference or not. All of a sudden, randomly, just airlines started to have middle seat vacant policy. But was it really helpful? Or is it just making people kind of feel good about any policy without truly having any impact on the infection spread? And we showed with our modeling work on the airplane that in fact that has nothing to do with the infection control. Middle seat vacant has no impact, literally, or very small impact.
What would you say, if you were to direct me one place to look for the best estimate of how much taking an airplane voyage increases your risk of over whatever else you'd be doing during those hours? Like, where would you refer me for that?
Well, I'm not a public health expert and I don't know all the detail, but what I can say is one thing is what we looked at it is the boarding policy. For examples, Southwest Airlines on one hand and American Airlines on another. Southwest has random boarding. Whosoever comes first goes in the plane, whereas American Airlines has zone-wise boarding. So we quantified if you take an airlines which has a zone-wise boarding versus a Southwest type of airline, how many infection that would generate. And you have to note one thing, in the airplane, infection is not spread during the flight because their ventilation system is quite good actually. The infection is spread during boarding and deplaning or during the waiting in the airport probably. And it's very rare also in the airport. So the question is to focus on these boarding policies. What we have recommended is take the flights in the Southwest rather than in the American Airlines, because zone-wise is creating more infection because people are waiting in their zones. And that waiting time is causing more contacts.
That's okay for me because I only ever arrive like at the very last minute. And they sometimes let me on and sometimes they don't.
So I have a question on the multi-patch model that you were talking about, because it's very interesting, like the number of parameters that there's interplay. Have you observed in the data that you've looked at for some regions that, say, for instance, the birth rates and the biting heterogeneity kind of match up such that it's very difficult to control, like one place blows up and then while it goes into a lull, the other goes up because of the movement patterns and how the host and vectors get replenished?
Well, we did analyze it mathematically. So I think we have not linked it with the data, data. What we were looking at, so parameter regime is quite complex, and we can divide into patches in the parameter regime that we can say one type of behavior will be in this patch, another type of behavior. So using these threshold parameters. So that was the task of it. If we can section this parameter regime, which is a complex, into these smaller parts where one type of characteristic or one type of threshold behavior exists. Now, the next step would be to use, if in some village, one situation occurs, then you pick up the result of that square piece where the parameter regimes are more feasible. So linking with the data is another level of work and require much more. Now, also the mathematical analysis quickly becomes very complicated. So we have talked about cases under different scenarios. So for example, we have taken a case where there is a dead end host. That means those are not transmitting the infection anymore. I mean, they are receiving the infection, but they are not, some hosts are not. So that reduces the number of parameters to a smaller parameter regime where you can divide parameter regions into small sections. And those section has the same threshold behavior.
Yeah, in fact, even for that 2-patch thing, if you map it out like a field and try to see like if there are there parameter settings where you actually get something like these loops, even in the mathematical characterization, it seemed like it's a very rich enough space that we might end up seeing the.
You are absolutely correct. It could have a very complex behavior. But remember, these are very well-defined systems studied and the way we are deriving this system, these have only 2 equilibriums. But the interesting part of the dynamics that we are seeing is not a forward bifurcation, but a backward bifurcation. And of course, there is likely that there could be a complex system, but from the literature, we know that we may not have other strange attractors in this type of system because we have extended the same work that we have seen mathematically proven in the literature. So the chances is less that we can see some periodic orbit or strange attractor in some sense. But it is, again, I cannot guarantee it because we have looked at only special cases of the system.
Thanks.
That's a good question. Yeah, thank you.
How much impact do you think climate change is going to have in the role of Chagas coming into the US and changing the vector?
A lot of changes. Actually, this is climate change changes the density of the vectors. And that is very, very important. But again, if you talk about in a short-term range, then climate change is an important aspect, and you can talk about seasonal outbreaks that are happening. But we are discussing on a long-term basis, and it may change some aspects of it. But again, we are doing this integral stability, which is a perturbation in the parameters. So basically, climate will going to change some insect-related parameters. And we have looked into the variation in these parameters, how that variation in the parameters will drive the system, how this dynamics will change. So we have given a comprehensive picture in that regard. But having said that, temperature and humidity has a major implications on a shorter time scale. Annual basis, for example, every year there is an outbreak because these are depends on density of the vector.
Okay, well, we're just past our stop time, so I just want to thank Anuj again for his great talk today, and thanks everyone for attending. Hope you can join again in 2 weeks for our next seminar.
Thank you, Anuj.
Thank you.
Great talk. We'll be in touch. Yeah.
Bye, everyone.